Hello students, find here handwritten important points of chapter number 11 Biotechnology Principles and Processes. Start with definition of biotechnology in general and definition given by European Federation of Biotechnology. Two core techniques that enabled birth of modern biotechnology. See the steps involved in construction of first artificial recombinant DNA molecule and steps involved in gene cloning process. Note three basic steps in genetically modifying an organism and then tools of recombinant DNA technology. Start with restriction enzymes. Then first restriction endonuclease. What is palindromic sequence? Nomenclature of restriction enzymes. What are sticky ends and blunt ends formed by the action of restriction enzymes on DNA? Specifically check the action of EcoR1 enzyme. After this, what are cloning vectors? Features of cloning vectors like presence of origin of replication, selectable markers and cloning sites. How selectable markers help in selection of recombinants and transformants. Insertional inactivation is explained here with the help of an example. Next tool is competent host. Check the technique used to make a host a competent host so that it can take up DNA. There are other methods also to introduce recombinant DNA into the host cell. One, microinjection, then biolistic or gene gun, and by disarmed pathogen vectors. Now, processes of recombinant DNA technology. Number 1, isolation of genetic material DNA. Number 2, fragmentation of DNA by restriction endonuclease. Number 3, isolation of desired DNA fragment by gel electrophoresis technique. Number 4, amplification of gene of interest by polymerase chain reaction and use amplified fragment to ligate with vector for further cloning. Number 5, insertion of recombinant DNA into host cell. Number 6, obtaining the foreign gene product in large scale by using bioreactors. And finally, number 7 step is downstream processing, which includes separation and purification of products. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.